So today it's just going to be 20 minutes, so it's going to be sort of a general overview of phospholipid fatty acid analysis and its uses. So I thought we'd start out with soil health and looking at how microbes are involved in the soil functions, and this is a slide I've used many times. So what I did this time was just sort of go over the functions and see if microbes are involved. So produce biomass, I think they're involved. You have, excuse me, you have mycorrhizae, you have rhizobia that help the plants produce more food and biomass. They regulate water flow and storage by creating flow through the soil. They help transform matter, though filtering is more of a physical property and buffering is more of a chemical property. They store minerals, they store organic matter, they support plants through the through mycorrhiza. And I think that if you look at past climates and vegetation and the ecology that's been there in the past, that can influence the microbes that are there now, and that could provide clues to the past also. So many of the soil functions are affected by the soil microbes. So I'm not sure what I clicked wrong here, but anyway. So when we were choosing the analyses, we were looking for physical properties, chemical properties, and biological properties. So we chose the physical, chemical, and much of the biological property analyses were fairly easy to choose, but we wanted a really strong soil microbiology analysis. So there were about 3 different kinds that we could choose. One involved cultures, so you had different kinds of media and you would learn about the microbiology based on which kinds of media had growth. But the problem is a lot of soil microbes don't grow on media. Another kind was involving DNA, and this was about 2012 to 2013, and a lot of advances have been made since that time. But at that point, DNA was more qualitative than quantitative. A really strong point is that it had the potential to identify to species or however far to a taxonomic unit that it could. One other thing about DNA analyses is that DNA has a legacy effect. It doesn't break down quickly in soil, so you don't know if it's today's microbes, yesterday's microbes, last year's microbes. Whereas phospholipid fatty acid analysis, the phospholipids break down pretty quickly. So they're strongly correlated to microbial biomass. So it's quantitative and it's estimating your living biomass, and it's kind of a snapshot in time. And because of this breakdown and renewal, it really can adapt to rapid changes in the environment. You cannot identify down to species, but we can identify into groups of microbes, and because it will get a list of all the phospholipid fatty acids and how much, you sort of get a fingerprint for that soil also. So that's what we chose. So what are phospholipid fatty acids? So I remember from being a college freshman, learning about what triglycerides are. You had a glycerol molecule and 3 fatty acids. So then if you replace one of those fatty acids with a phosphate group, you have a phospholipid fatty acid. So here's the important part. Most cell membranes have a phospholipid bilayer. So if you can extract the phospholipids from a soil, you're going to get a good estimation or a good indicator of how many microbes are in the soil, because the more microbes, the more cell membranes, the more phospholipid fatty acids you have. The other part about phospholipids is the fatty acid tails are not all the same. They're different lengths. Some of them have double bonds. Some of them have 2 double bonds. So there's all different ones, and some of these fatty acids will indicate different types of organisms. So I'm just going to go through the organisms, the groups, the main groups of organisms that you can identify or categorize with phospholipid fatty acids. You can divided into gram-positive and gram-negative bacteria. And I get asked a lot what is meant by that. So this was actually a staining process that was developed by a man named Graham. And he found that some bacteria would stain with crystal violet. And it turned out that was because they had this thick layer of peptidoglycan above in the plasma membrane, or above the phospholipid fatty acids in the cell envelope. But there were other bacteria that did not take the crystal violet stain. So they would counterstain with safranin. So you'd have purple bacteria and pink bacteria. And it turned out that the gram-negative bacteria had 2 layers of phospholipid fatty acids, just a thin layer of the peptidoglycan. So with PLFA, the gram-positives have branches on their fatty acids, whereas the gram-negative bacteria have a single double bond or maybe a 3-carbon cyclic portion on their fatty acid. So the gram-positives are overall resistant to stress, and they're found throughout the soil profile, where the gram-negatives tend to be found more in the rooting zone, and they're more sensitive to water stress. Now, Actinobacteria, they're indicated by a methyl group on the 10th carbon of the chain. So for phospholipid fatty acids, that's how Actinobacteria are indicated. So they're the ones that produce the smell that you relate with, that you identify with soil. They're filamentous bacteria. We used to think they were fungi. They grow throughout the soil, and they're actually gram-positive bacteria and act in some ways a lot like them. For arbuscular mycorrhiza, there's a 16 carbon fatty acid that has 1 double bond at the omega-5 position. So these arbuscular mycorrhiza, in exchange for the carbon that they get from the plant roots, they extend the plant rooting area and help the plant bring in more water, more moisture, more nutrients such as phosphorus and nitrogen. Soil fungi are indicated by an 18 carbon fatty acid that has 2 double bonds. And the fungi, one of the main things they do in the soil is they help to break down the resistant cellulose and lignin substances that are added to the soil as plants and trees die. Other eukaryotes are indicated by fatty acids that are polyunsaturated, which means they have 2 or more double bonds. So in this group, you have protozoans, nematodes, insects, earthworms, acari, crustaceans. The protozoans are indicated by the 20 carbon length fatty acids, and the other eukaryotes are indicated by other polyunsaturated fatty acids. So one of the important things that this group does is they are the upper end of the food chain, so they prey on the soil bacteria, and because they have a lower carbon to nitrogen ratio, as they devour the bacteria, some of the nitrogen is released into the soil and made available to plants. Okay, so just to give you an idea, that there are these different groups, but they all have multi-functions within the soil. It's one giant web. I think this was designed for looking at the protists. You'll see that some of them feed on bacteria, some of them feed on fungi. They're predated upon by other arthropods and nematodes, but they can also be parasites. Other organisms can be parasites to them. So just a lot of interaction found at different trophic levels throughout the soil. Okay, so just to talk about our experience, we selected phospholipid fatty acid as the analysis that we were going to do and we purchased the equipment and the software and training in early 2013 from MIDI Incorporated. And we chose the high throughput method of the extraction. It had been a 4-day extraction, but Jeff Beyer came up with a method that took 2 days using the 96 well sampling trays. So it's a high throughput method. We're a service lab, so a lot of times we don't have that much information about the samples run. We do cooperate with University of Missouri researchers and some other researchers where we sometimes have information about the samples. So we primarily run research samples. We tend to discourage individual samples because it's an expensive analysis, and an individual sample is difficult to interpret, and there's a lot of information that's given, and so it's difficult to explain that to an individual without something to compare it to. We've run about 15,000 samples from about 30 different states and 3 different countries. We've analyzed samples for nationwide studies for 2 different studies through NRCS, Soil Health and Soil Science Division, and we're getting ready to start another one. The high throughput PLFA, just so you have an idea of what all has to be done and how long it's going to take. We take subsamples. Usually people give us larger samples than what we need, so we take a representative soil sample and we freeze dry it or vacuum dry it and then freeze it. And then when we extract it, it's a 2-day process, and after it's extracted, we run it on a gas chromatograph, and that takes another 25 hours. So then the data needs to be transformed, downloaded, formatted, and emailed to the recipients. So the software lists the individual PLFAs, the total PLFA, then it classifies it into microbial types and fat types, and we, in the last couple of years, we also have ratios that are calculated, and they can be useful also. We have some quality control measures. We include a blank and a standard in every run to confirm that the batch was extracted properly. We freeze dry and vacuum dry the samples for a couple reasons. We have consistent sample weights that way if we dry them all down to the same amount, and also we feel it helps with the extraction because you don't have soil moisture to dilute the extractant. We use an internal standard in the extractant, so it's a known amount of a PLFA that's not found in nature, so we can use that. We know how much should be in every sample, and we can use this for corrections that, through the process. So looking at the results, the column in the gas chromatograph, what it does, it's sort of like, it's not really what you think of as a column. At the University of Missouri, we have all kinds of columns here that represent the university, but this is more like your intestines. So what it's doing is it's separating the phospholipids. The smaller ones go through first. So you get a peak, and where the peak comes off helps to identify what fatty acid that is, and then the size of the peak or the area underneath it indicates how much of that phospholipid fatty acid was available in the sample. So the results report that we provide, it has 7 pages. The first page is one that's used probably the most out of any information. It lists all the different phospholipid fatty acids that were found in the sample, and then it's given as in a molarity, a picomoles per gram of soil. So that total PLFA, that's what correlates strongly with the total microbial biomass. We do also give just the number of phospholipids that were found in there. That's just something to know. It's not a super important thing to report, but just so you have more information. We also give all the phospholipids that were found as a relative abundance. This isn't used as much, but it can have uses. Probably the second most common use is we give the totals that were indicated for the different microbial types based on the indicator phospholipid fatty acids. So for most of these, more is generally better, especially for AM fungi, gram-negative bacteria, eukaryotes, and fungi. But you do have to remember that there's the possibility of these being pathogens. Another way that we give this same information, but it's as a percentage of the total, so it's relative abundance. So this is more like making a pie chart. What is the relative abundance of all these different kinds with the total population considered as a whole? So we give the information how all the different phospholipids were used to categorize by microbe. We also give these categorized as fat types. This is not used quite as much as the organism types, and a lot of this is sort of redundant, like the 10-methyl that indicates the Actinobacteria. There's some new information given here, and those can be used also. Again, we give the information how all the fat types were categorized. And then the ratios, this is a relatively new feature, but it's used, I think it's pretty useful. Let's say you have, if you're looking for differences, you may have trends with the fungi. Maybe one treatment tends to have higher fungi and it tends to have lower bacteria counts, but not significant. If you look at it as a ratio, you may be able to find significant differences. So most of these fungi to bacteria, predator prey, saturated, unsaturated, the bigger the better. I generally consider the gram positive to gram negative to be, the smaller the number, the better. And again, we show how all the ratios are calculated. I would like to say that besides just the PLFA information, it's also very helpful to have other data so you can see what's going on in the soil that might be affecting the microbes. So it's good to get other data. Things to know about PLFA: it was not developed as a soil health analysis, but it offers more biological information for the overall assessment. People spend their whole careers on PLFA, so it's a pretty steep learning curve. I'm learning things all the time. It's a lot to get going on. I don't believe there are concrete, good, across the board, good or bad values if you're looking at total PLFA. It varies so much, which we'll talk about in a little bit. You do need to know that archaea is one kind of organism similar to bacteria that do not have PLFA, so you're not going to be able to look at those amounts. Comparisons are very important. The analysis is expensive, but if you look at the price per unit data point, it's very reasonable, and it's okay. So this was just how we give the people the information of how we categorize the fatty acids into fat types. So the ratios, it's just a different way of looking at the data. It's a newer feature that they've provided with software updates. So what I like is if you were looking at, if you had a trend where a certain treatment tended to have less fungi, but it didn't come out as being significantly different, and maybe the same samples tended to have lower bacteria, that might not come out as significant, but maybe if you look at the ratio, maybe it will. So overall, most of these ratios, the bigger the number, the better, except for the gram negative. Generally, I like to think the more of the gram negatives that you have, the better. So in this case, it would be the smaller, the better. And sometimes it's just a way of looking at it. I'm not really sure about the mono and poly. There's different things you could look at that. But again, we provide the information about how the ratios were calculated. Other data can be useful, so it'll give you information, maybe why some of the microbes were coming out as they were. If it's a really low pH, maybe that's why the fungi was more dominant in those. So anyway, just other data can help you out in looking at what's going on with these microbes. Things to know about PLFA: it was not developed as a soil health analysis, but it offers a lot more biological information to the overall assessment. People can spend their whole careers concentrating on PLFA, so there's a really steep learning curve. There's a lot for us to learn in the beginning, and I continue to learn something all the time. I don't believe there are concrete across the board good or bad scores or values. Archaea, this is something you need to know, they do not have phospholipid fatty acids in their cell wall. So this is one microbial group that we can't use this to evaluate. So other things, comparisons are really important. It's expensive, but if you're a researcher and look at the price per data point, it's really very reasonable. You get a lot of information for what you pay. There's many factors that affect soil PLFA. You have chemical factors, pH, the level of nutrients, parent material, mineralogy, soil taxonomy. Is it a young soil, an entasol, a mollisol? Soil texture, the density, weather and climate. So overall temperature versus temperature and soil moisture at the time, management, tillage, soil amendments, time of year. If people are going to make comparisons, we suggest they take all the samples at the same time of year, from year to year and within the year. And it varies quite a bit with soil depth. The microbial content of soil drops rapidly with depth in soil. So some of the limitations of PLFA: it only gives an indication of who the microbes are, the general groups. It doesn't identify the species, doesn't tell you what the microbes are doing, or give you their activity level. It does provide relative biomass. There are conversion factors differing for if you want to figure out the biomass of fungi or bacteria. We generally suggest that if you're reporting, you just report the values that we give, otherwise you're introducing area, but it would be interesting to look at that. We suggest that there are a lot of references. Some of the most important is the high throughput fatty acid analysis that we use. There's use and misuse of PLFA measurements in soil. That's good to look over. Wikipedia is a good place to start. There's a nice article there about phospholipid fatty acids. And Dr. Kristen Beam, who works for the ARS here at the University of Missouri, she went with us to be trained in this, and she has written up a paper that will discuss what you can do with the data that we provide. So just some points. An overall one, PLFA analysis is a tool to use in determining the effects of a treatment on soil microbes. And because of all the variables that affect PLFA, I don't believe at this point that it's the type of test that you're just going to use and it doesn't tell you exactly what you should do to adjust your soil microbes. So I think in some cases it may not be what say a farmer may be looking for, but I think researchers can use this to determine what effects the treatments are having on soil microbes and use that information to advise farmers.